Welcome to another lecture by Medical Medics, Immunology Chapter 5, Antigens and Antibodies. In this lecture, we will focus more specifically on antigens. What makes a good antigen? Furthermore, what are antibodies and what are the functions of these antibodies? We will talk about classes of antibodies, also known as immunoglobulins. We will then talk about antigen-antibody interaction, the clinical relevance of antibodies, and end with a summary. Now, first of all, what are antigens? Antigens are molecules capable of triggering an immune response by binding to immune receptors. Now, the sources of these antigens can be pathogens like bacteria, viruses, fungi, parasites. It can be non-infectious molecules like toxins, allergens, or transplanted tissues. We also have the concept of self-antigens. These can trigger autoimmune diseases when misidentified as foreign. So essentially, let's say we have a bacteria, right? This pathogen, in this case a bacteria, okay, carries what we call antigens. This antigen is capable of triggering an immune response by binding to immune receptors. So this cell, let's say it's a neutrophil or another immune cell, has receptors. These receptors will bind with this antigen. And when they do, we will have different responses and reactions occurring. We also learned that this antigen can be found in several pathogens like viruses, parasites, etc., but also in non-infectious molecules like allergens or toxins. We also come to know that we have self-antigens, whereby for some reason our immune cells have linked up with an antigen that is actually not part of some pathogen, but rather belongs to a friendly cell, so to speak, something that belongs in our own body. However, the immune cell that attaches to this self-antigen doesn't know that this belongs to our body. So the consequence, which is the immune response, will be the same. So whatever destructive consequences could follow from a immunological response, an inflammatory response, or whatever, will still be the same. It's just the cause is from the self. And this is essentially what we study in autoimmunity and autoimmune diseases. Now, following the discussion on antigens, we have mainly two types of antigens. We have immunogen or immunogenic antigens, meaning an antigen that elicits an immune response. But we also have hapten, which means a small molecule that becomes immunogenic when attached to a larger carrier. So all in all, in layman's term, you can think of antigens as ID badges, identification badges that cells or germs or pathogens have that our immune system uses to recognize and then respond to it. Now, what makes a good antigen? Well, let's look at the properties of immunogenic antigens. First of all, foreignness, meaning it must be recognized as non-self. That's the first property. The second one is the size. So larger molecules are more immunogenic. The complexity. Proteins are better antigens than lipids or carbohydrates. And then dose and route. So the amount and method of exposure can affect the immune response. Essentially, the immune system reacts more strongly to complex, unfamiliar invaders. And hence, the idea is to elicit a strong immune response. That is what makes a good antigen. So what properties does this antigen need? It needs to be able to be recognized as non-self, so being foreign to us. The larger, the better, due to higher immunogenicity. Complex proteins makes it react even more. And then dose and route. So the amount of it and the method of exposure all play key roles in the response. Now then, what are antibodies? Antibodies, also known as immunoglobulins, abbreviated often as Ig, are Y-shaped proteins produced by B cells to bind specific antigens. And if you remember, antigens are molecules capable of triggering an immune response by binding to immune receptors. And what are then the structures of these antibodies? Well, we have a variable region. So if you look at the illustration right here, you can see the antibody illustrated, and the variable region is the tip part of the antibody. So this part in purple. These are the arms, so to speak, that bind to the antigens. And these are specific antigens, right? So we call this bit, the arms that bind to antigens, the variable region. Then we have the constant region. And this is the stem that interacts with other immune cells. And it is colored here. So just like the name implies, this is constant. And this is, let's change that. And this tip here is variable or varies. The upper part, the tip, is specifically binding to a specific antigen. So they will look different between all antibodies. The constant region is what communicates with other immune cells. I can add that it is also here in the constant region where we identify which type of immunoglobulin, which class of antibody, so to speak, that we are talking about. And since antibodies and immunoglobulins mean the same thing, this is where we identify the different classes. So for example, IgA is a type, IgE is another one, IgM, and so on. In coming slides in this lecture, we will talk about the most relevant ones, so to speak. But keep this in mind. The antibody has this Y-shaped structure. It's a protein. The antibody is produced by our B cells. It is made of two main regions, the variable one and the constant one. The constant one is where we can identify the class of the antibody and where communication with other immune cells occur. The variable region, the upper tip, is where we have specific bindings with specific antigens. And what are the types of bonds that happen? Well, antibodies bind antigens through non-covalent interactions like hydrogen bonds and ionic interactions. And then what is the functions of these antibodies? Well, first of all, neutralization. So it prevents pathogens or toxins from entering the cells. There is opsonization, whereby it coats pathogens to make them easier for phagocytes to engulf. Further, we have complement activation. So it triggers the complement cascade to help lyse pathogens. Another one is antibody-dependent cellular cytotoxicity, meaning helping natural killer cells to destroy infected or abnormal cells. So antibodies, as you can notice, act like multitasking tools. They are blocking invaders, they are tagging them, meaning marking them. They are then communicating information to other immune cells to help destroy them, etc. So they are very multifaceted and interesting structures. Now then, we mentioned classes of antibodies. And if you remember, the class of the antibody or the type of immunoglobulin was identified in the constant region. Exactly. We also had the variable region. This is where it binds with specific antigen. Now, what are some of these types of immunoglobulins? Those you will likely come across throughout your studies and medical career. First of all, immunoglobulin G. This one is the most abundant antibody in the blood. Furthermore, it provides long-term immunity and crosses, this is very high yield, it crosses the placenta to protect the fetus. Next to it, we have immunoglobulin A. This one is found in mucosal surfaces, like in our gut or respiratory tract, but also in secretions, like saliva or in tears. Furthermore, we have immunoglobulin M. This is the first antibody produced during an infection. It forms pentamers for strong binding. Another one is immunoglobulin E. This one is involved in allergic reactions and defense against parasites. Furthermore, we have immunoglobulin D. This one is found on immature B cells and are involved in early B cell activation. So different types of antibodies have specialized roles, just like a team with different players. So to make it a bit more relevant to clinical practice, like we said, IgA found in mucosal surfaces, right? A deficiency, therefore, in IgA could lead to what? Well, it can lead to recurrent respiratory and GI infections. What about IgD? IgD, which is involved in what? Early B cell activation. And you should already have an idea from the previous lectures about the important role of our B cells. This is where we get our memory and our antibodies from, right? The B cell activates into an antibody factory. And we've discussed now what antibodies do. What about IgE? Well, this one is crucial and key in allergic reactions. So we will see elevated IgE in, for example, asthma or eczema or anaphylaxis, etc. We can also mention IgM. This one is the first antibody, right? The first antibody produced in infections. High levels indicate a recent or acute infection. Now, I do not encourage you to memorize too much right now because we will repeat all of these over the course of the duration of this course. However, what you should pick up on is that we have, first of all, antigens, we have antibodies. Antibodies have a classification, and this is discovered or pointed out in the constant region of the antibody. This is where we have M, A, G, E, D, etc. All these different ones have different roles. So there are different players in the same team. We have also discussed the important role of antibodies. That are several. First of all, blocking invaders, marking them, alerting our immune system, getting help, so to speak. So if you have an idea right now with something like this, that's very sufficient for now, and we will just continue to build up on that. But let's just briefly repeat. Antigen-antibody interaction, because that's what it's all about. So two key concepts. There is specificity and there is cross-reactivity. So each antibody binds only to a specific antigen. And the specific part of the antigen that an antibody recognizes is called the epitope. So let's go back to the basics just for a brief overview. We had some bacteria entering our body. We have neutrophils, right, reacting to it. We have then macrophages coming here, engulfing and breaking down this bacteria. This macrophage, or the dendritic cells, etc., we call them antigen-presenting cells. And the clue is in the title. It's an antigen-presenting cell. So it's going to break down, pick up the specific antigen belonging to this bacteria and put it on its own surface to express it to the rest of our body. So it's going to present the antigen. Then, coming back to what we're talking about now, is that we have these immune cells that are going to identify this. If we have antibodies produced, let's say this is an antibody. Excuse the drawing, but this is an antibody, okay? It has specific receptors, right, to identify this specific antigen. And the part of this antigen that is presented, the specific part of the antigen that the antibody here is meant to recognize, is called the epitope. Now, this is more a drawing illustrating a re-exposure because we have antibodies produced, right? The first time around, when we have an invader, we have the innate immune system responding to it. That then, afterwards, presents the antigen. Our T cells invite and activate our B cells to do what? To start producing antibodies with this specific variable region, meaning the specific receptor that will target this antigen in the future. And so upon re-exposure, now we have these antibodies floating around. And as they are floating around, they will recognize the epitope and immediately respond to it. Now, the second concept, which is crucial here, is cross-reactivity. So sometimes antibodies bind to similar antigens, leading to unintended reactions. And this is extremely important because we have a whole field of study, which is autoimmune diseases. And it is precisely from this kind of area that it stems from, meaning that once we've understood the specificity of recognizing these antigens, we then have to ask ourselves, we have an extremely safe system in place for cell proliferation, right? If we go back to basics, we have cells that become two daughter cells and then they keep proliferating. But just like we learn that we can have mutations in our cells, we can have errors occurring whereby we have overproliferation of cells. Something has gone wrong, either with the system that's meant to stop it or just the cell itself and the nature of that mutation. So we get overproliferation of cells. Eventually, we arrive at the development of tumors. And this is where we talk about the field of oncology, so cancers. Meaning that cancer is a consequence of something normal going wrong, which is also why studying physiology is so fundamental. So you understand how it's supposed to be, to then learn, and even, I would say, as a good doctor, you're supposed to be able to assume things going wrong. And how would that look? If you understand how it's supposed to be, it will make it far more clear to you when things do go wrong. And it's the same here. The system is supposed to identify very specific epitopes, but there can be errors. You may have receptors on cells in your body that are harmless, that are crucial to your overall functioning, that share 99.9% similarities with this epitope. It is not inconceivable that the antibody attaches to that one mistakenly. And it doesn't know, it doesn't understand that I just attacked the wrong target. So what happens is that the cascade that follows from an antibody attaching to the epitope will still be the same. We will still have the inflammatory response. And it is the response that many times is what causes destruction, disease, the development of illness, etc. So what is the overall clinical relevance of antibodies? Well, we use it in vaccines. So we stimulate antibody production to prevent infections. Essentially, we introduce part of that antigen we want to produce antibodies for. We give that to the person in a small dose, enough to get our immune system to react to it without causing harm. That's the idea. And then the immune system will behave the same way as if it was a real threat. It will identify the antigen, have it presented to it, and then start producing via our B cells a lot of antibodies for this specific antigen. And when we do that, we now say that this person has been vaccinated, meaning that if in the future the actual real pathogen with that antigen enters into our body, we already have antibodies ready to attack it. Furthermore, we have allergy testing. So this is where we measure IgE levels to identify allergic responses. Autoimmune diseases. So autoantibodies attack the body's own tissues. For example, rheumatoid arthritis, lupus. Furthermore, we have monoclonal antibodies. These are engineered antibodies used in cancer therapy and autoimmune disease treatment. And we will dive into all of this and in greater detail as we move along. So in summary then, antigens are molecules that trigger immune responses. And antibodies are proteins that target these antigens. This one is worth writing down. Furthermore, antibodies have multiple functions, including neutralizing pathogens, tagging them for destruction, activating other immune responses or processes. Furthermore, the immune system tailors antibody response to fight specific invaders effectively. So, in a simplified way, you can say that antigens are the bad guys, and antibodies are the immune system's tools to find, mark, and to stop them. And that's the end of this lecture. Go on now to chapter 5.